Dear students, and today we are going to continue the same section, the photosynthesis, and see the first question. Stomata of cam plants. Okay, so there are choices about their role and position in the particular plant system. Always open during the day and closes at night, open at night and closes at day, and never open. Okay, first you should know something about the cam plants. So what do you mean by cam over here? So cam refers to crassulacean acid metabolism. Okay, so it's a kind of photosynthesis system happening in some kind of plants. So normally these cam plants are present in a very dry conditions. Okay, for example, the cocktail plants. Okay, so they belongs to the particular cam or crassulacean acid metabolism plants. So over here they have to adopt the particular dry condition. Okay, this is what you have to remember. Okay, so as we all know, what is the role of the stomata in the leaves over here? So what is stomata first? Stomata are the openings present on the plant surface, that is leaves. Okay, so there is a structure called aperture and it will be guarded by two guard cells. Okay, so they will open and close based on a plant system. Normally they are always open during the daytime and always close during the night time. Okay, so why they are always open and during the daytime? Because through the particular opening they have to absorb the particular carbon dioxide and they have to release the excess of water during the photosynthetic process. Okay, so this is what you have to remember about the stomata. Stomata is the opening in the leaves. Okay, and they will always open during the day and they will always close during the night. This is the normal thing you have to understand about the plant system. But as I told you, over here in case of the cam plants, okay, so I told you they are in a very dry condition. Okay, so they have to adapt to the particular dry environment over here. So they have to prevent the loss of water from the particular system. Okay, to prevent the loss of water, they have the adaptation. Okay, over here, based on that their stomata will function completely differs from the particular normal stomata. Okay, so over here see the choices. Never open is a completely wrong answer. They have to open at some time, then only they can do the particular carbon dioxide assimilation. Okay, so always open is also should be a wrong, because if they are gonna be always open, they'll lose excess of they lose excess amount of water and they cannot absorb the particular carbon dioxide. So both A and D are wrong choices. Now you have to choose between the B and C. Open during the day and closes at night. Open at night and closes during the day. Okay, as I told you, they have to adapt to the dry condition. They have to prevent the water loss during the sultry weather. So the answer choice over here is they are always open at night and closes at during the daytime. Okay, so this is how you have to understand the cam plants. So what kind of phosphorylation possible in the cam plants? As I told you, there are two types of phosphorylation. We have seen C3 and C4. Over here normally they will go for C4 kind of phosphorylation, C4 kind of carbon dioxide assimilation. Why? They have to get more, or they have to assimilate more amount of carbon dioxide to increases the biomass by using the available amount of light energy. Okay, and see the second question over here. Which of the following absorbs light energy? Okay, and there are choices for you: ribulose bisphosphate, oxygen, water, and chlorophyll. Okay, we'll see one by one. Ribulose bisphosphate is an acceptor molecule. We all know that. Okay, they are going to accept carbon dioxide. This is what you have to remember. Ribulose bisphosphate is an acceptor molecule. We all know. Okay, this is a 5-carbon structure, 5-carbon organic molecule. They are going to involved in a Calvin cycle, that is C3 cycle, and they are going to accept carbon dioxide, not the particular light energy. Okay, so light energy means over here from the sunlight the light will be traveled as in the form of photons, the packaged form of photons, and the photons will be absorbed by some form of structure. That is what we are going to see now. And the second choice over here is oxygen. Okay, so just remember one thing, oxygen over here in case of photosynthesis, oxygen is being released from the particular system. It is not going to be absorbed by the particular system. Normally the oxygen will be released or evolved from the photolysis of water. Okay, so the photolysis of water leads to the release of oxygen plus release of electrons plus protons. Both electrons and protons going to enter into the electron transport chain, and the further photosynthesis system and synthesis of ATP and NADPH molecule, and the oxygen will be evolved from the particular system. Okay, so both the oxygen and the ribulose bisphosphate are the wrong choices over here. Then you have choices chlorophyll and water. Okay, so you may get confused with the choice water. Okay, because as I told you, the very first step in the photosynthesis over here, the photolysis of water. So you may think that the water may absorb the light energy. That is not a correct choice, because water is being split over here by means of the energy utilized by the particular system. Okay, so what is the source of the particular energy? Energy is from the light source in the form of photons, and the photons energy form will be absorbed by some kind of photosystems, and the photosystem made up of the pigments called chlorophylls. Okay, the chlorophyll will absorb the particular light energy in the form of photons. They got excited, and the excited form will split the particular water molecule. After that, they will release the particular oxygen molecule, and the electrons will be transferred to the further system, and the transfer of electrons will form the ATP and NADPH molecule, and then the formed ATP and NADPH molecule will be utilized for the synthesis of the carbohydrate. This is what the sequence of the process in the particular photosynthesis. What is the very first process? It starts from the light source, photons, and the absorption by the pigment system, chlorophyll, and the chlorophyll will lead to the excitation of the P700 or P680 system based on a photosystem 1 or photosystem 2, and from this only the electron transfer going to happen. And before we are entering into the electron transfer, it will split the particular water molecule, and that will release the oxygen and other atoms over here. And see the third question over here. Select the correct statement of the following. Okay, so one or two statement may be correct over here, and you have to read the statement and then you have to answer the particular type of question. Okay, and there are four statements given for you. Set scheme of light reaction takes place in presence of PS1 only. Okay, so to understand the first statement, you should know something about the set scheme. So what do you mean by set scheme? So as we have seen in our previous classes, there are two systems going to involved in the photosynthesis, PS2 and PS1. Okay, so as I told you in a previous question, light, then photocenter, P680, and the excitation, the electron acceptor, okay, then electron transport chain, then again next sender, photocenter. In case of the PS1, the electron excitation, electron acceptor, then the formation of NADPH, and over here in case of electron transport chain, the formation of ATP. Okay, so this is what the sequence of event we have seen it now, and here you can see the shape, the Z shaped reaction. Okay, so that is why we are calling this as Z scheme. Okay, and the question, the statement over here, the Z scheme of light reaction takes place in presence of PS1 only. You can easily see it over here, involvement of both PS1 and PS2. So the first statement may not be correct, because as I told you, here the statement says PS1 only. This is wrong. Both PS1 and PS2 involves in the Z scheme. And see the second statement. Only PS1 is functional in case of cyclic phosphorylation. So we have seen in the previous classes, there are two types of phosphorylation possible in case of photosynthesis, cyclic and non-cyclic phosphorylation. So what do you mean by non-cyclic? So this system is non-cyclic. So over here the electrons will not be transferred back to the system. They will be finally transferred to the acceptor molecules, and they will synthesize the particular ATP and NADPH molecule. Okay, so there will be no cyclic or rotation of the particular electrons over here in case of the particular non-cyclic phosphorylation. But in case of cyclic phosphorylation, only the PS1 system gonna involve. Over here the electrons will be cyclic and they will synthesize only the ATP molecule. Okay, this is what you have to remember. In case of cyclic phosphorylation, there will be no synthesis of the particular NADPH because the particular enzyme required for the synthesis of NADPH is absent over here. Okay, so that it will leads to synthesis of only the ATP molecule, and the electrons will be transferred back or cycled back to the particular system. That is why we are calling this as cyclic phosphorylation. Okay, and see the statement, only the PS1 is functional in the cyclic phosphorylation. This statement is correct. And see the third statement over here, the cyclic phosphorylation results in ATP and NADPH2. As I told you over here, the enzyme NADP dehydrogenase is absent in the cyclic phosphorylation, so there will be no synthesis of NADPH molecule in the particular cyclic phosphorylation. Only the ATP molecules will be synthesized in the particular PS1 and cyclic phosphorylation. So the statement over here says cyclic phosphorylation results in ATP and NADPH. NADPH will not be synthesized over here, so you can easily eliminate the statement as wrong. And see the next statement, stroma lamella. Okay, stroma lamella, stomata lamella lacks PS2 as well as NADP. Okay, so this is what the statement given for you, and you have to remember one thing, the cyclic phosphorylation. Okay, so they are going to happen in a stroma lamella. That means they don't need the particular PS2 system because the cyclic phosphorylation going to happen in a particular area. What is stroma lamella? In case of the particular organelle, the chloroplast, the structure, the membranous protruding structure is called stroma lamella. So over here, in case of the stroma lamella, they lack the structures called PS2 and NADP because, as I told you, only the cyclic phosphorylation going to happen in the stroma lamella. They don't require the particular PS2 and NADP because they are not going to synthesize the NADPH molecule. They are not going to involve the non-cyclic phosphorylation. So this statement is correct. Okay. So what are the statements correct? Only the 2 and 4 is correct. Okay. So where is the 2 and 4? The first one is correct and the remaining are the wrong choices. Okay. This is why you have to understand the particular type of question. There are statements given for you and you have to read the statements very thoroughly and you have to understand the concept of the particular statement. Based on that, first you have to choose the correct statement, then you have to see the choices, and among the choices you have to choose the correct combination of correct statements, then only you have to answer the particular type of question. And see the next question over here. The electrons from excited chlorophyll molecule of PS2 are accepted first by. Okay, so this is what the clue for you. So over here you have to note down over here the system. They are talking about the photosystem 2 and they are talking about the electrons and they are talking about the accepting molecule, the first accepting molecule. Okay, so see the choices over here, cytochrome B, cytochrome F, ferrodoxin and quinones. Okay, so as I told you, you have to carefully note down over here, this is talking about the PS2 system. So as I told you, there are some differences between the photosystem 1 and the photosystem 2. Okay, so in case of photosystem 2, over here the reaction center will contain the chlorophyll molecule P680. In case of PS1, they contain the particular system P700 chlorophyll system. Okay, so over here the sunlight energy absorbed in the form of photons. Okay, and then the molecular excitation will happen and the excited molecule will releases the electrons and the electrons will be accepted by. Okay, this is where the question arises over here. The accepting molecule from the particular photocenter, after the molecular excitation, the excited molecule will releases the electrons. So over here in case of photosystem 2, okay, so you have to remember one thing, the cytochromes are involved in the electron transport in the in between process. We have seen the set scheme. Okay, so the electron transport happening in between the PS2 and PS1 over here, or in case of cyclic phosphorylation, it will happen within the particular PS1 system, but it is an intermediary process, not the very or initial stage of the particular photosynthetic reaction. Okay, so the cytochromes are involved in the intermediary process, that is the electron transport chain. So you can easily eliminate the cytochromes B and F, and the remaining two choices over here, the ferrodoxin and quinone. Okay, so in case of the photosystem 2, in case of PS2, you have to remember one thing, the first electron acceptor groups are the quinone molecules. Okay, plastoquinone or phytoquinones. Okay, they are gonna accept the electrons from the excited region. This is what you have to remember. Ferrodoxin is not a right answer. I will explain you why. Okay, so quinone is the correct choice for the PS2 electron acceptor. And see the next question. This is a continuation of the first question. In PS1, the first electron acceptor over here. As I told you, you can easily eliminate the cytochromes and plastocyanin. They are the some other pigments involved in the photosynthesis, and again you have the choices among the two, ferrodoxin and iron sulfur proteins. Okay, just remember one thing, both the ferrodoxin and iron sulfur proteins are involved in the PS1 system. As I told you, the ferrodoxin is not going to involve in the PS2. Okay, ferrodoxin also going to involve in the PS1 only. Okay, but the question over here is the first electron acceptor. Okay, so the first electron acceptor in case of PS1 is iron sulfur proteins. Okay, this is what you have to remember. Whenever you have a choice of the iron sulfur proteins, you have to choose this answer as a first electron acceptor. When you don't have the particular iron sulfur protein, then you can choose the ferrodoxin as a electron acceptor for the particular PS1 system. And again I will remind you, you are not supposed to choose the ferrodoxin as an electron acceptor for PS2 system, because ferrodoxin is only involved in the PS1 system. Okay, so in case of the particular PS2 system, as I told you, from the excitation, the electrons will be accepted by the next system over here as, okay, so over here it will be accepted by the iron sulfur protein in case of the PS1. And the iron sulfur protein is a iron containing protein. Okay, it's a iron containing protein. So over here it will accept the electrons, then it will transfer the electrons to the particular ferrodoxin. From the ferrodoxin it will be transferred to the downhill energy level, and that will produces the ATP and NADPH molecule over here. Okay, so the point you have to remember, in case of the PS1, it will accept the electrons by means of the iron sulfur proteins. And see the next question over here, in case of the photosynthesis, energy from light reaction. Okay, energy from light reaction to dark reaction is transferred in the form of, okay, so as we all know, there are two major reactions, light and dark reaction. In case of light reaction, the energy molecules will be produced and the energy molecules will be transferred to the dark reaction. In the dark condition, they will fix the carbon dioxide to the carbohydrate molecule. Okay, so the point over here, in which form the particular energy is being transferred. Okay, so we all know that the ATP and the ADP molecule may be the correct answer, and you may get confused with the ribulose bisphosphate and chlorophyll. Okay, chlorophyll you can easily eliminate because it's a pigment, it is not gonna transfer the energy. It is gonna excite the molecule by absorbing the light energy, but they cannot transfer the energy, so you can easily eliminate the chlorophyll. This one is ribulose bisphosphate, this one is adenosine triphosphate, adenosine diphosphate. All the three components having the phosphate group, okay, they all can carry the energy, the electrons, okay, energy in the form of electrons. But the question over here, which form the energy is being transferred from the light to the dark reaction. So over here you can easily eliminate the ribulose bisphosphate because they are going to involve in the accepting of the carbon dioxide molecule. They are not going to involve in the transfer of energy from one step to another step. And you have the choices of ADP and ATP. ATP is being synthesized from ADP, but you cannot transfer the energy in the form of ADP. Okay, I will explain why. Adenosine, this is adenosine diphosphate, this is adenosine triphosphate. Okay, so this phosphate group is far from the particular adenosine. Okay, so you can easily break the particular phosphate group from the particular adenosine molecule. So the breakage of this molecule will release us the energy, and that energy will be utilized by the other reactions of the particular system. Okay, so that is why we are calling the ATP as a energy carrier molecule. You cannot store energy. Just remember this statement. You cannot store energy. You can carry the energy in the form of the ATP molecule. ADP molecule you cannot use as an energy carrier. Okay, as I told you, in case of ADP only the two phosphate groups are there. Okay, the energy required to break the particular phosphate group from the ADP will be very high. So you cannot use the particular ADP as a energy carrier molecule. So the point you have to remember over here, always you can transfer the energy in the form of ATP, not the ADP. Okay. So ADP required to synthesize the ATP. Okay, as I told you in the previous classes, ADP along with the inorganic phosphate, they are going to synthesize the ATP by using the proton gradient in the process electron transport chain, or the phosphorylation happens in the electron transport chain during the particular photosynthesis and in the respiration. We are going to see the respiration later in the classes. So the point you have to remember over here, for the particular type of question, in the photosynthesis, energy transfer from light to the dark condition over here in the form of ATP. Okay, this is what the answer. And see the last question over here. The first carbon dioxide acceptor enzyme in C4 plants. Okay, so this is talking about C4 plants. Okay, this is the first clue for you. And carbon dioxide acceptor molecule, this is the second choice, second statement or clue given for you. And over here you have to note down one thing. This is talking about enzyme. Okay, this is what you have to note down very carefully. Okay, this is talking about C4 plants, talking about carbon dioxide accepting molecule and carbon dioxide accepting enzyme. This is not talking about the molecule, acceptor molecule. This is talking about acceptor enzyme. Okay, so over here there are choices given for you. Ribulose diphosphate or ribulose bisphosphate is a carbon dioxide acceptor molecule. Okay, ribulose bisphosphate or ribulose diphosphate is a carbon dioxide acceptor molecule, organic molecule, 5 carbon molecule. They will accept the carbon dioxide and they will become the intermediate 6 carbon molecules, and they will produce 2-3 carbon molecules. Okay, so you may tend to choose the particular ribulose bisphosphate as an answer, but this is not a correct answer. As I told you, ribulose diphosphate is a molecule, but over here you have a clue, ribulose diphosphate carboxylase. Okay, but you have to remember one thing, ribulose bisphosphate gonna involved in a C3 plants. Okay, this is the acceptor in the C3 plants, and ribulose bisphosphate or diphosphate carboxylase and rubisco both are same enzyme. Okay, so rubisco is the ribulose bisphosphate carboxylase. Co refers to carboxylase and oxygenase. Okay, so both are same enzyme. So this is a kind of activity of the particular rubisco enzyme. Okay, so both the enzymes gonna involved in the C3 plants, not in a C4 plants. And remaining two choices, phosphoric acid and phosphoenol pyruvate carboxylase. As we have seen in introduction classes, in case of C4 plants, the primary acceptor of carbon dioxide is phosphoenol pyruvate, and that molecule, that reaction is being catalyzed by the enzyme phosphoenol pyruvate carboxylase oxygenase, that is PEPCO, phosphoenol pyruvate carboxylase oxygenase. Okay, so the answer over here for the particular type of question is PEPCO or the phosphoenol pyruvate carboxylase. Okay, so the complete name for the particular enzyme, PEPCO. So why we are calling this as PEPCO? Phosphoenol pyruvate carboxylase oxygenase. Okay, that is why we are calling this as PEPCO. Okay, so the carbon dioxide, first carbon dioxide acceptor enzyme for the particular C4 plant is PEPCO. Okay, this is what the answer. In the next class, we are going to discuss something about the respiration in plants. Okay, then we can going to discuss the question we can expect from the particular topic.